It's a great pleasure to be talking to you today, and I'm talking about a topic that should be of interest. Sex, gender, and the brain should be of interest to a lot of people, not just neurologists. And my talk will be a 2023 update because I have given this talk a number of times, both in the United States and internationally. And every time I give it, I have to rethink what I'm doing because things have changed in the meantime. And I have nothing to disclose. And the big change this time compared to previous version of my talk is that in 2022, the National Academies of Science, Engineering, and Medicine published this report, which is shown in this slide. This report is the most recent attempt of reaching a consensus on issues such as sex, gender identity, and sexual orientation, which all are complex, interrelated, and distinct, and extremely difficult to measure. So the big change today is that I'm trying to align my language to be almost 90% aligned with the Academy of Sciences, which will trickle down to NIH, FDA, CDC, all the way down to you for your next grant application. So I think it's extremely important, the topic. These are the learning objectives, which you have probably already seen. So my presentation has three pieces, and then as a conclusion. The first piece is sex. Sex, I will talk about chromosomes, hormones, and reproduction. Then I will talk about gender, and things get more complicated. I will distinguish two different chapters of gender, a subjective or personal identity side and the social, cultural, and political side. Then I will try to explain to you why these sex and gender are important to you as medical researchers at the Mayo Clinic. And so I will take a disease as an example. I will use dementia and show you how you can apply this sex and gender thinking and conceptual frame to look at causes of dementia in the population and how these can actually influence the way you think of preventing the disease or intervening. I will also try to, because this is a QHS seminar, to give something technical about the difference between frequency and magnitude of an effect, which is simple, but actually quite dramatic. And then I will draw some conclusions. It is clear that when I talk about sex and gender, I am simply trying to summarize to you things that I have read or listened to or learned. I am not an expert of sex and gender. So I'm not talking about from personal experience, but when I talk about dementia and Alzheimer's disease, that is where I have done work myself in the field. Okay, I don't like when people talk about things that they have not done because they tend to not talk correctly, but so is life. So let's talk about sex. Sex is extremely important, and you will see my presentation is a lot of sex, just because sex to me is the most relevant to biological research in medicine, but it's not the only thing. Sex is very understandable to us that are trained in biology, medicine, nursing, whatever science. So sex has to do with chromosomes. There are chromosome complements, XX and XY, you all know. They distinguish male from female. These chromosomes are responsible for the development of the gonads. The gonads are the ovaries and the testicles. Some people prefer the term testes instead of testicles. I don't like testes, it's testicles. So do what you want. And these hormones produce, these gonads produce hormones. The hormones are estrogen and progesterone, primarily in women, and testosterone, primarily in men, but not only, as you will see soon. Things are quite complicated. The way humans reproduce is such that female are the only one of the two that are able to have a pregnancy, and so they experience pregnancy, breastfeeding, and they cannot experience, as of today, oral contraception, which has primarily been a female intervention rather than a male intervention. So let's jump into the biology. This is a very fascinating area, and this is very scientific, so there is nothing political here. The problem is that until last century, and when I say last century, I feel like laughing because I was like many years ago, that's when I was born. So anyway, I'm from last century. Last century was the 1900s. In the 1900s, the science was very backward. And they used to believe that all of the sex differences in non-gonadal traits, such as fat distribution, hair distribution, muscle distribution, were all mediated by the gonads. So there was a very simple thinking. You have XX or XY. This will determine whether you develop testicles or ovaries, and the ovaries and the testicles produce a lot of hormones, and the hormones will make you different. Well, that is completely limited, because the new version of the new century, which is the 2000, is that there are two pathways from which you can go from the chromosome to the sex differences. And that is because, in addition to controlling the development of the gonads, the X and XY and XX complements can have direct activities that are chromosomally mediated that do not go through the gonads. Let's try to understand what that means. And of course, I am quoting a lot of the work done by a very famous biologist at the UCLA in California, Dr. Arthur Arnold, who is, I think, one of the pioneers of this new way of looking at sex in biology. So you are in front of a dimorphic trait, let's say distribution of fat in men and women, or in male and female, and then you ask the question, is it gonadal or is it not gonadal? If it is not gonadal, then which chromosome is responsible? And which genes on which chromosome are responsible? If it is gonadal, then you can say which hormone is responsible and at what age or in which period of life this has happened. Let's go through step by step. First of all, these are the chromosomes. These are very nice, simple picture. Two chromosome X, quite big and similar. Of course, they are homologous. In the male, you don't have the second chromosome X, and you have this baby chromosome Y, which is a little bit of a dwarf there. And this chromosome Y is very special because it has a piece that is homologous to chromosome X. So it behaves like a pseudo-autosomal. And then you have a piece that is called the SRY region, or sex region on chromosome Y, which is the piece of chromosome that is responsible for developing the testicles. If you don't have that gene, you will develop ovaries. It's not correct to say that it's by default, but you can say simplistically, by default, you have ovaries unless you have the SRY gene. This is completely limited, but let's go with that. And then you have a piece of chromosome Y that is completely specific to male and does not have an equivalent on chromosome X. What you immediately see is that it's impossible to imagine having double dose of all of these genes in female and being compatible with survival. So the way nature goes is that one of these two chromosome X is inactivated or blocked so that it does not express itself. But things are a lot more complicated than this. If you go to the map of chromosome X, you will discover that things are a lot more complicated. First of all, we are talking about 1,700 genes, and these genes are in different families. In red, pinkish purple, you have the genes that can be repressed or inactivated. In dark blue, black, you have genes that are pseudo-autosomal, meaning that they have an equivalent on chromosome Y, so they behave like a normal pair of genes. And then you have the one in green, which escape inactivation. These are genes that should be blocked, but they are not blocked, so the women become carrier of two doses of certain genes that are not on chromosome Y. This is called escaping inactivation, and you will see why this is important. The problem is that chromosome X is extremely crucial to control a lot of functions, not only in the brain, which is what we like to study, but also in cancer, cardiovascular, cell regulation and metabolism, endocrine system, immunology, and reproduction. So the fact that you could have double dose of certain gene or gene escaping inactivation or different expression is tremendously important in diseases and prevention. So let's see what are the three most important mechanisms by which a male and a female can have different genetic expression from chromosome XX or XY. These are the two complements right there. And here you have the female, and here you have the male. There are three mechanisms. One is due to the chromosome Y. Why? Because men have a piece of chromosome Y which is not homologous and is only in male. And this chromosome can then express themselves, of course, protein, enzyme, and structure, or they can influence other genes everywhere else in the genome through gene-gene interaction. So this is unique to male, the female cannot do. But female have two mechanisms alternative. One is that they have a double dose of all of the genes on chromosome X. And even though they block majority of these double dose, some escape inactivation. So the female have two doses of some genes that can be extremely important for diseases or lack of diseases. The second mechanism that is typical of women that men cannot have is having paternal and maternal X chromosomes. As you know, a male can only have a yellow or maternal derived chromosome X because it has only one chromosome, where female can have paternal in blue and maternal in yellow. And so, depending on which one of the two is blocked or not blocked, the other one will express. And depending on that, you will express the genetic variant that came from your father or your mother, because you have the option of doing that. And this does not happen all or not, it's not black and white. It happens in a way that multiple cells and different tissues have different percentages of switch, paternal, maternal, which makes women mosaics. Instead of being a single type of cells, women are a mosaic of paternal, maternal X expression. I will talk a lot about this, which is crucial. How can we study the fact that you can have a gonadal and a non-gonadal mechanism? Well, the group at UCLA has done incredible work in the last 15, 20 years, where they said we can manipulate mice. So now I'm talking about mice, not female and male humans. In mice, you can create transgenic models, and you can create 4 families of mice. You can have mice that are XX, so female, and they have the normal ovaries. That's nice. There's like classic female mice. Then you can have male mice that have XY and they have testicles. So M means testicle, F means ovaries, XX means chromosome for women, XY means chromosome for men. However, because they could take the SRY region from chromosome Y and take it out and implant it on an autosomal chromosome, they can have this genetic region coding for the testicles in a different chromosome. So you can have mice that are XY, like in female, like in male, but with ovaries, because you remove the SRY gene, so they develop ovaries. And then you can transplant the SRY gene on an autosomal, and you get a female with testicles. So you can create, I call them monsters. These are mice that are chromosomally male or chromosomally female, but gonadally the opposite. Do you see that? This can be done in mice through genetic manipulation. If you now start looking at these 4 families, you can learn a lot of things about whether a trait is gonadal or non-gonadal, of course. And that's what the group in Los Angeles has been doing. Look at body weight. This is a very important variable in metabolic research, diabetic research, and all of that. Look at what happened. If you look at mice after development on day 75, the male are heavier than female. So the XX, the male with testicles are heavier than the female with ovaries, regardless of their chromosomes. So at development, the gonads are driving and decide that being M makes you heavier than being F. These are testicles and ovaries. But if you remove the gonad on day 0, it takes 7 days for the situation to completely flip. And by day 7, when there are no more gonads, the mice that are XX are heavier than the mice that are XY, regardless of the gonad that they used to have before removing them. So that means that in a normal development of mice, the gonad drive the show, but when you remove the gonad, the chromosome drive the show, and they do it oppositely, where XX is heavier than XY. But then the question is, what is the problem? Is it having 2X that is making me heavy, or having a Y making me light? Well, the next experiment addresses this question. They took other transgenic mice and they created 4 families of mice. X0, like only one X, XY, we used to call them male, XXY, male with an extra X, and XX, we used to call them female. You follow that? This can be done by genetic manipulation in the laboratory. Well, look at what happened when you remove the gonads. that the group of mice that are carrying 2 doses of X shift heavy, and the group of mice that carry only 1 X shift low, regardless of how many Y they have. Y is irrelevant, because having 1 Y here does not make a difference, having 1 Y here does not make a difference. What makes a difference is having 2 X's. So they discovered that the mechanism is probably a gene on chromosome X escaping inactivation, causing females to have a double dose of that expression if they have from 2 X's, regardless of the gonad. Do you see what I'm doing? So this is becoming a full field of research. There are people doing this full time in laboratory around the world, trying to understand the non-gonadal causes of sex differences. Fascinating. Another fascinating phenomenon is the chromosome X inactivation, which we already have faced. This slide is a courtesy of a colleague from our department, Dr. Stacy Winham. She developed this very nice pedagogic cartoon. So imagine that you have 6 cells of a tissue. This could be the liver, the brain, the kidney, the lung, whichever tissue you like. And you have a maternal X and the paternal X. The maternal is red, the paternal is blue. And then you can block one of the 2 by inactivation. The inactivation is done biologically through methylation and other ways of blocking the gene expression, primarily methylation. But look what happened, that you can block the maternal or you can block the paternal. So the other one will be expressed. So if you do transcription and translation, then you will get the RNA will only be the one that can be transcribed, and then the protein will be the one that comes from that RNA. So you have a complete mosaic situation. In this tissue, you have 3 paternal and 3 maternal, but the percent of paternal maternal can range from 0 to 100. You can have all paternal, all maternal, or 80% in between. So your liver can be from 0 to 100 paternal maternal. This is revolutionary understanding the fact that the female body can dose itself where the male is on. And of course, the male can only do one business, which is copying the X from the mother and go on with that. So if you imagine that the father and the mother carry 2 different variants of a gene that is predisposed to a disease, your probability of having the disease depends on what mosaic percent you have in your organ. Fascinating. This has been demonstrated by many groups. The best study, which I like very much, is from Johns Hopkins University. They did something simple and extremely magic. They colored the chromosome X of the father mouse in red, and in green, the chromosome X of the mother. So they make the 2 mice, and when the little mice came out, the litter, they look at the female mice, and they discovered that they were not either red or green, which would mean that the change happens very early in the development, and then the full clone of cells is either green or red. No, it happens very late in the development, is regional, is patternal, and is lateral, it has lateralization, which is completely unexpected. Look at this. This is the brain of a female offspring of this colored gonadal father and mother. This is the right side, this is the left side. Not only there are mapping of red and green all over the brain, but the left side is more red than the right side. This has been found in the retina, in the kidney. All of the pair organs seem to be asymmetric in terms of mosaic expression. We don't know why, we don't know what controls this expression, and the mice of the litter were all different from each other. None 2 sisters were the same. Fascinating. But let's go to the gonad, because this is the old-fashioned last century, our century story, and we like, of course, this because we have heard it before. So now the question is, which hormone and when? Okay, let's take the life of a human being and divide it into periods from pregnancy, intrauterine life, all the way to death in different colors. And on the y-axis, I'm going to show you the amount of circulating hormone as a percent of the maximum attainable in either set. Okay, so assuming that the maximum amount of hormone circulating in a human, male or female, is 100%, I'll show you the percent. Very interesting. Look at testosterone in women. Testosterone in women is very high intrauterine, goes down to 0 at birth, is flattish all the way through puberty, goes up a little bit and stays up, not much compared to what you will see in men, but it's there, and then it tapers away. Apparently, even though this testosterone is limited in amount, it's very important to control sexual behavior and mood in women. Let's look at the driver of female hormonal situation is the estradiol. Estradiol is extremely important for women. Look at the amount intrauterine, almost maximum, down to 0. In the first half year of life, there is a peak. This peak is extremely important because it will tell the body, get ready to become female or become male. So the first half year of life is crucial to prompt all of the cells of the body to become one way or another during puberty. Then you have a silence period or a latency. And then with puberty in the women menarche and the appearance of cycle, you have gigantic increase very rapidly in women. And then you reach the maximum production. It depends on where you live, but between 10 and 15. And then you have the maximum amount of estradiol fluctuating, of course, every month, all the way to age about 45. And then you have a precipitous decline all the way to the end of life, which is almost as dramatic as the menarche. Men have a completely different story. Men, of course, have a lot of estradiol. It's very important in men. It's high during intrauterine life. It goes down to, everything goes down to 0. The newborn is almost hormone neutral. There's a little peak on the first year of life. If this peak is too high, it causes a big mess. So it has to be a little peak, but not too big. You will see the real one of testosterone. Then there is nothing happening in childhood, very little. And then in puberty, slowly, there's an increase of estradiol. It's not very steep. And then it stays very high all the way to the end of life. The beauty of this is that after age 65, a man has more circulating estradiol than a woman. Yeah. Anyway, and then, so this is fascinating. But look at the testosterone. Testosterone is magic, very high intrauterine, goes down to 0. There's a huge peak in the first year of life. If you don't have this peak, you will never be a man. It's what determines the prompting of the peripheral cell to go in the direction of masculinity versus femininity. Goes down to almost 0. With delay compared to women, you have puberty, but it's 2-3 years later, as you know very well. Reaches the maximum somewhere around 15, 16, 20. And then it stays quite high, but starts declining soon, but decline very smoothly and slowly down to the end of life. You see what's happening in this picture? How the sexes, gonadally mediated effect are age dependent. And there's an incredible interaction between sex, hormonal sex, and age. Let's see then what happens. Well, females have 2 major revolutions in their endocrine system, the menarche and the menopause. These are like a complete shift of their hormonal setting, which dictate changes in metabolism, energy production, all kinds of things. Men have only one major change, which is puberty, and then the rest is primarily slowly going down. This has tremendous repercussion because these hormones are not only controlling. In last century, people believed that the sex hormones were important to make the endometrium develop, to develop the breast, to develop the testicles. That is completely misunderstanding because there are receptors of estrogen and progesterone in all of the cells and tissue of the body. There are almost no cells without receptor for testosterone, estrogen, and progesterone. So that means that all of these tissues and organs, call it the brain, the bone, the liver, the lung, the kidney, the heart, are under endocrine control of sex hormones. This is the big revolution of this century again, similar to the gonadal and non-gonadal, is the fact that the sex hormones are not sex hormones, are hormones, like the thyroid hormone, the adrenal hormone. You understand? It's complete misunderstanding of thinking the sex hormones are sex hormones. Sex hormones are hormones. It seems to be simple, but it actually blocked research for 20 years. This not understanding of the endocrine effect of the gonads. Because they thought that the gonads were only good to make ova and sperm. That was complete misunderstanding of the gonads. Anyway, women in the human species have this magic ability to reproduce. They are the ones that reproduce. Men only participate in reproduction as donors of sperm. So the women have hormonal cycles, as you know very well, and therefore they can have hormonal contraception, which is based on blocking the cycle or altering the cycle. Women can experience pregnancy, and pregnancy can have positive effect on the body. There is a full literature on the positive effect of having very high level of estrogen during pregnancy. But as you look a little bit nearer, you will see that there is high level of estrogen and progesterone, but the estrogen is not estradiol, but have to be primarily estriol, which is a weak agonist. So there's a full lecture to be given to what happens in pregnancy, but we cannot do it today. There are adverse events in pregnancy, like hypertensive pregnancy disorder is the classic group of symptoms that can be very problematic in women because they can lead to proteinuria, diabetes, hypertension, preeclampsia, eclampsia, all the way to convulsion and death. So the pregnancy can be an incredible positive event or an incredible stress test for the body of the woman where things start showing. There are important exchanges between the fetus and the mother. This is a recent development in the last 20 years. Not only can you exchange genetic information like RNA, DNA, but now we know that there are complete stem cells moving from the fetus to the mother and populating the tissue of the mother. So after a pregnancy, a mother becomes a chimera or a microchimera. A chimera is a Greek mythological entity where you have an animal that is half an animal, half another animal. So you can have a goat and a lizard. You can have a man and the bull. I mean, it's a mixture of two animals, and it's a classic of the Greek mythology. So women that have pregnancies are microchimera, and it has been shown that you can detect these cells in their tissues after the pregnancy. This is a full area of research, and it's developing right now. It's fascinating. Anyway, then women can breastfeed and women can experience menopause, as you have seen. So far, it was only sex. Now we jump into gender, which is a complete other kettle of fish, as they say in London, because people say, oh, gender is cultural and societal. Well, that may actually be partly incorrect, more complicated than that. Gender has two components. There is a subjective or self-perceived gender, which is called also gender identity. This is the self-declared feeling of being a male or a female. It's self-reported. And this then as a gender expression, because depending on this feeling, a number of people decide to act on this identity through behavior, appearance, so the way they talk, the way they move, the way they dress, the way they comb their hair. So this is all of the expression of gender identity. Also, there's a full area of psychology studying personality, masculinity, femininity, psychological style, personality. All of this is a full field of research that has to do with the expression of gender identity. This is becoming a big field because it has been progressively more recognized that there are people who have a gender that is non-congruous with the sex that was declared at birth. Because human beings, when they are born, at the time of delivery, a nurse or a doctor of the family look at the genital and say, oh, it's a boy, oh, it's a girl. Well, this very often is simple, sometimes it's not simple. There are examples of ambiguity, which is a full other lecture, which I'm not giving today. But assuming that you got simple genitalia, then you are a boy or a girl. Well, this is called sex declared at birth and get into your birth certificate. But later in life, you may discover that you feel that you are the opposite sex, which becomes gender, and you become a transgender to be distinguished from a cisgender or non-transgender. There's a big debate on whether people that are non-transgender are non-transgender or are cisgender. And there's a full debate in the book that you should read on this, and there's still disagreement. I don't like cis-trans, but that's the terminology. So if you write a grant, you need to be aware that that's the way they are called. Then, depending on the sex declared at birth or the gender that you declare having, regardless of the sex that you were given by the nurse or by your mother, then society will respond to you through legal, cultural, religious practices, so that in some societies, being a male or a female changes whether you go to school or you don't go to school, whether you can have certain jobs, you play certain sports, you can have access to medical care. And in some society, being a boy or a girl involves manipulation of your body, such as circumcision, genital manipulation, genital mutilation. So society takes the decision to change your body following societal, religious, or anthropological practices. This is an incredibly important situation where it's not that the biology that drives the gender of the society, but the society modifies the body, called opposite control. And of course, in certain countries, women cannot drive a car, they cannot vote, they cannot own properties, and many of those kind of things. So this is the domain of gender. As you can see, gender is worth a month of study by itself, because there are many, many components into gender. Gender is not as simple as gonad, non-gonad. So sex is complicated, but it's a piece of cake. Gender is way more complicated. Are you following? Now, so what? Well, let's take all of these pieces and see what does it mean if I do research on cardiology, dermatology, gynecology, choose your field. Well, I'm taking dementia as an example, one because it's a good one, and one because I know it very well. And I'm now going to try to tell you how my thinking of sex and gender gets applied to research in biology at the Mayo Clinic today. I am doing some simplification, of course. I mean, my talk is a full oversimplification of things, but 45 minutes is 45 minutes. With some oversimplification, I will give you 4 possibilities of ways in which sex and gender influence the risk of dementia. So we are dealing with risk and protective factors, and we are looking at 4 possibilities. One is that male and female have the same frequency of exposure, but they have different effect or magnitude of the action. And I will use an example from genetics, which is the ApoE4 gene. There are other genetic examples, which I don't have time to do today. Then I do the opposite, a scenario where they have different frequency of the exposure, but same effect. And I will use education. I could use smoking, alcohol, but I don't have time. And then we are going to look at examples where a factor is both of different frequency and of different strength of activity when you have it. And I will use traumatic brain injury. Another good example would be HIV infection, but I don't have time to do it today. And then we see 2 examples of factors that are restricted to only one sex, and these are bilateral oophorectomy and androgen deprivation therapy. So I'm going to go now through each one of the blue, highlighted in blue, examples to show you how sex and gender all of a sudden becomes important in understanding dementia. The first example is same frequency, and I'm using the ApoE4 allele. As you probably know, ApoE4 is a risk factor for dementia, and it sits on an autosome, so not on the sex chromosome. It's actually chromosome 19. So it behaves like Mendel, okay? So it's Mendelian inheritance. If you believe in Mendelian inheritance, which you may also decide not to believe, but if you believe, the probability of inheriting a variant from your father and mother on an autosome is the same. Brother and sister have the same probability of inheriting ApoE4 because it's an autosomal chromosome. It's not X or Y. Well, if you inherited the variant and you are a female, you have a bigger magnitude of the effect of that gene on the risk of dementia or Alzheimer's disease, so that the component of dementia which is attributable to that gene is bigger in female than in male, even though the frequency of carrying it is the same, because it's the response of the body to having the gene that is differential by sex. Do you follow this? And when we talk about magnitude of the effect, in epidemiology, we talk about relative risk, odds ratio, hazard ratio. This is an illustration of what I was just trying to tell you from 1997. Look at what happens to people that carry 2 genes, two doses of the ApoE4, which is the harmful variant. women have higher risk than men quite clearly throughout a large part of life. And if you look at carriers of a single gene, it's even more dramatic. If you have a single dose and you're a female, you have more than double the risk of dementia than if you have a single dose, you're the brother of that girl, and you carry the same gene, right? This now has been, of course, confirmed many times, and now we know that the explanation is that there is an interaction between ApoE4 and estrogen in the control of the accumulation of proteins in the brain, such as amyloid and tau and vascular lesions, so that having an ApoE4 in presence of estrogen is different than in absence of estrogen. This is an example of sex effect, which is hormonally mediated, so it's a gonadal effect, we think, for the moment. I'm simplifying a lot of things. Now, let's see the opposite. Now, we have different frequency, but same effect. So, we are postulating that if you send a boy and a girl to school, their brain will develop similarly. Okay, so we take an equalitarian concept of biology. If you send boy to school, they will become smart. If you send girl to school, they will become equally smart. Why is then a problem low education? Well, because last century, the probability of a woman to have high education was very low compared to a man, because the political, social concept was that men were the breadwinner, they were one having career and education, women were staying home, taking care of the house and of the children. This has been true all the way to almost the end of last century. It changed quite a bit after World War II. So if you study people that are young, old now, you will discover that there is more dementia attributable to low education in women than men, not because the brain of women is not good if you educate them, but because they were not educated for political, historical reasons. And now, things have become equalitarian at the end of last century, and now they are flipped the other way around. So now in the United States and many European countries, women are educated more than men. So in 30 years, this will be completely flipped the other way around. Something like that has happened for smoking and other things. So you see, in this case, there is nothing biological. This is social, this is political, this is historical. This was a social decision to not educate girls. And this has nothing to do with biology, but yet it will cause the brain to be less protected against dementia, because being educated creates mechanisms of protection of your cognitive abilities. And when you age better if you have been educated than if you're not been educated. But this is not biological, this is social political. Another example which makes things more complicated, we are cranking up another level of complexity, is when there is both frequency and effect difference. I think the majority of factors are actually more of this type. You have two components. Let's talk about traumatic brain injury. And let's talk about traumatic brain injury in young people and adults, because things are different in children and elderly. But in the midlife, in the big chunk of life, men tend to have more trauma than women, and they have different trauma. Because as you know, men and women, you know that men do contact sport, combat, they do jobs like construction, road construction, they are a little bit crazy, they drive crazy. So men have different behavior than women and different social roles, which cause them to be exposed to traumatic brain injury. Okay, men have more injury than female, you accept that. Well, things are complicated because if a man and a woman are sitting in the same car and in the same car accident with the same lesion, the woman will be better than the man. So the men have less ability to respond to trauma. Their brain is more sensitive, or women are resilient, so that you have a combination of more frequency and more susceptible. So this causes that dementia attributable to traumatic brain injury is bigger in men than women for two components, higher frequency and higher effect. This has been shown beautifully by a recent study done in US veterans. These are military veterans of the United States. Look at what happened of the risk of dementia over age. If there is no traumatic brain injury, the continuous red and black, these are people without injury. You can see that men and women have the same risk of dementia with age. This has been shown again and again in the United States and Canada. We do not see a difference in risk expressed as incidence between men and women. This is not the frequency of cases of dementia, which has to do with the numerator of prevalence. This is incidence or risk. So we don't think there is a difference in risk of dementia by men and women. However, if you have a traumatic brain injury, the dotted line, assuming that on average the severity of the trauma is equal in men and women, you need to make an assumption of equality of severity, then the risk of men is higher than women, and is significantly so, suggesting that men are more prone to respond negatively to the trauma by developing dementia, where women are protected in some way against the insult. Do you follow this? Beautiful. These are pedagogic examples coming out in the literature every year. This is coming out now. We are starting to understand the profile of what's happening. The two more extreme examples, pedagogic, are only one sex, oophorectomy in women. If you remove the ovaries to a woman before menopause, you will have an increased risk of dementia and Alzheimer's disease. This is only happening in women, of course. So that piece of dementia is only attributable to a sex-unique factor. Our group has been working a lot on this. We are probably one of the biggest groups in the world working on this question. These are some of our data. If you look at the risk of dementia and cognitive impairment in the referent women in black, you see this beautiful conversion risk increase with age. If you look at women that have oophorectomy later in life, when they are postmenopausal or perimenopausal, there is nothing. But if you do the oophorectomy younger, you have an increased risk, which is significant, suggesting a 60% increased risk of dementia. Do you see how this is an example? This piece of dementia is only attributable to the ovaries, and the men don't tell. But the men have testicles, as we mentioned before, so you have the similar mirror image of this. If you treat men with androgen deprivation therapy to protect them from prostatic cancer metastasis, which is very common, unfortunately, it's being done today at the Mayo Clinic, these men are protected from dying of prostate cancer, but they will develop more dementia, because of course the block of the testosterone has similar effect to the block of the estrogen. This has been shown now beautifully in a number of studies. Not all studies are aligned. I'm showing one that is pedagogic. There is some debate about this. But this is a study published in 2019. Instead of doing the cumulative incidence, they did the survival free, which is simply flipping the curve. And the men that did not take the blocking of the androgen did better in terms of dementia than the one that did take the drug that blocked their androgen. Do you see this? So, similarly, one gonad off problem, the other one gonad off problem. So very, very interesting understanding of it. So, what are my conclusions? My conclusion is that it is very important when you study dementia or any disease of aging to look at a life course approach and a dimorphic approach. Dimorphic means that you have a path for male and the path for female, and that all of the risk and protective factors that will modulate the pathway to positive aging or to disease aging are interacting with sex and gender at different ages. So you have an age, sex, gender interaction in predisposing people to dementia or keeping them protected against dementia. And this new way of thinking will completely revolutionize medicine, will be all rewritten. It will take 10, 15 years, then all medicine will be rewritten in dimorphic terminology. Because sex is biological, it involves chromosome, gonad, and reproduction. Gender is social, cultural, and political. Sex and gender variable affect aging and diseases. All diseases are sex dimorphic. Dementia and Alzheimer are very different in men and women. The risk factors are different in men and women. We saw ApoE, education, traumatic brain injury, and we saw some factors that are only single sex, like oophorectomy and androgen deprivation. So my conclusion is that we need to move toward a dimorphic neurology. Actually, I suggest to move toward a complete dimorphic medicine, and we rewrite our textbook of medicine with two different volumes for male and female. Thank you very much.